So I'm going to be talking about Natera, and we are not just a non-invasive prenatal diagnostics company. We've been offering diagnostic services for several years in various clinical domains that have to do with reproductive testing. So I'm going to talk at a very high level. I was told this talk had to fit into just 8 minutes, and there's a lot more data, so please, if you have questions, there'll hopefully be time to address them afterwards. The concept behind Natera is that you have all of this amazing signal processing and computer technology in your gizmos today, what we put in our laptops, what we have in cell phones, what we put in spaceships, and very little of that signal processing and computer capability has found its way into diagnostics, especially diagnostics for reproductive indications, which is bizarre because there is so much room for improvement, as Ken mentioned, and the most important thing in most people's lives is not the latest gizmo, it's having a healthy family. So the way Natera goes about this is we look at the data from the Human Genome Project, and we look at the latest, greatest signal processing techniques, and we apply that to improve the sensitivity and specificity and coverage of diagnostic testing, specifically for reproductive indications. So there's been a whole range of tests that we've introduced over the course of the last few years, and our involvement in non-invasive prenatal diagnostics can be seen as the culmination of a whole series of tests that have led up to this. We started off several years ago offering the first microarray to analyze a single cell for aneuploidy across all 24 chromosomes, simultaneously being able to detect deletions, duplications, and mutations at multiple genetic loci. So this is now being used by several hundred IVF centers around the world. We've tested about 40,000 embryo samples at this point, and it's used by families who are trying to avoid genetic diseases in their children. So, for example, a family that has a risk of cystic fibrosis or carries a BRCA1 mutation, as well as by couples who want to just improve the probability that embryos implant during an IVF cycle. Then we were the first company that offered a SNP microarray for analyzing products of conception. The idea here is about 10% of pregnancies will miscarry at a stage late enough that you might have a product of conception which doctors would be interested to analyze. And there are two reasons why they do this. The one is you might have systemic chromosomal abnormalities like balance translocations, which will cause a miscarriage, and you want to be able to address those systemic chromosomal problems if you can. And the other is if you find that a patient is suffering from recurrent pregnancy loss, but there are no chromosomal abnormalities, then a whole series of additional tests are warranted in many cases. So this is now covered by insurance. We are in contract with insurers that cover about 60% of the patients who go through our products of conception test. And the idea here is you can do a test in just 2 days. Instead of waiting 3 or 4 weeks for the cells to be karyotyped, you don't have to culture the cells. You get a very rapid result. We are not affected by maternal cell contamination to the extent that we can detect the maternal cell contamination, which affects about 20–25% of samples that go through a regular karyotype. And we can detect things at a much more fine-grained level than you can typically see with a karyotype. And then we were actually the first company that did high-throughput analysis of free-floating fetal DNA in the mother's blood. And while all of our trials were going on for the non-invasive prenatal diagnostic, we launched this non-invasive prenatal paternity test. And this is a very interesting market because about 5 to 10% of pregnancies around the world, the father on record is not the true father. So until we came along, a woman who was pregnant and wanted to establish paternity would have to either get an amnio or CVS, which is obviously an invasive procedure which carries a risk of miscarriage, or they would wait for the child to be born. Now with this test you can basically do a blood draw just 9 weeks into the pregnancy, and we can do a paternity test which is very reliable, 99.99% plus. So this has been launched with the leaders in paternity testing around the world. Our main distributor in the United States is DNA Diagnostics Center. They do about 500,000 genetic test samples a year, which is more than LabCorp and Quest. And then internationally, we've got relationships with a lot of labs and phlebotomy centers all over the world. Then most importantly, we are currently under multiple clinical trials, one of which is funded by the National Institute of Health for the non-invasive prenatal diagnostic. And the key thing here is clinical coverage. We focus on all 5 chromosomes, namely 13, 18, 21, X, and Y, and simultaneously we do high-throughput carrier screening. So the idea of high-throughput carrier screening is about 1 in 500 pregnancies is affected by trisomy 21 or 18 at birth. about 1 in 500 pregnancies is affected by an inherited genetic condition like cystic fibrosis, Tay-Sachs-Canavan's disease, etc. So it really makes no sense to have the world's best test for Down syndrome if you're not simultaneously testing a whole bunch of diseases beyond cystic fibrosis, which are just as relevant to the health of the pregnancy. One of the key things is that over time, rather than offering services out of our CLIA lab, we are translating our tests to run on the desktop sequencing systems, because there's really been a revolution in sequencing capability in the last couple of years. The power of sequencing is evolving at a rate about 3 times faster than Moore's law in terms of the number of sequence measurements you can make for a certain cost. And the sequencing capability is now placed on these desktop sequence systems, which are about the size of a large printer. So we envisage a world in the long term where these desktop sequencing systems are going to be placed at hospitals and labs all over the world. They will run the samples themselves, and we will run the informatics in the cloud as a web service. So here's an overview of how the technology works. I'll first talk about this as it applies to single cells or trophectoderm biopsies that come from an embryo about 5 days after fertilization. And then I will talk about how we apply this to non-invasive prenatal diagnostics. So when you get a single cell in the lab, we amplify up the DNA. And because you're just looking at a single cell, every position on the chromosomes that you're looking at is just a single molecule. So you get very high rates of allele dropouts, where a molecule's there that you can't see, or drop-ins, where you think a molecule's there that really isn't. And amplification biases upon different parts of the chromosomes. So if you just look at that data quantitatively, using a CGH array or using quantitative sequencing, you make a lot of mistakes. What we do differently is we take data from the Human Genome Project, and we also take data that you can directly measure or infer from the parents. And from that a priori information, we can work out where the chromosomes crossed over in the sperm and the egg that fertilized to become the child. And if you can work out where those crossovers happened, you can basically reconstruct the embryonic DNA in silico. And so we can turn those very noisy direct measurements into reliable copy number calls across all 24 chromosomes and simultaneously call mutations at multiple loci. Now, the same approach conceptually is applied in the non-invasive prenatal context, except here, our data is not coming from a single cell or a small clump of cells from the trophectoderm. Our data is coming from many hundreds of copies of the fetal DNA, which are existing in the mother's plasma. And we identify the SNP locations, which will be maximally polymorphic, which will give us the most information about variations between the mother and the father. And we will sequence those loci preferentially using a multiplex PCR assay, which targets those particular regions. And we're able to multiplex 11,000 PCR primers in a single reaction, which I think is to my knowledge, there's nothing out there which can replicate that level of multiplexing. And more than 90% of the reads that come out of that highly multiplex PCR reaction are valid reads that map to the genome. So with that information, we will then evaluate a whole series of hypotheses. So at a high level, your hypothesis will be, did I get 2 chromosomes from the mom, 1 chromosome from the dad, like a maternal trisomy? Or did I get 1 chromosome from each parent, disomy? And then within those broad hypotheses, we're looking at a huge number of, literally billions of sub-hypotheses which say, where did the chromosomes cross over in the parents to generate these chromosomes that I see in the child? So for every hypothesis, you generate the probability of that hypothesis, given the measurements that you've made on the plasma and what you can infer about the parents. And you take the maximum likelihood probability as your diagnostic, say trisomy. But now you have a probability associated with that hypothesis. So you know the confidence in the diagnostic that you've made for that particular sample. So if we do a blood draw very early in the pregnancy, say 8 weeks, and we have a tiny fraction of fetal DNA, we might say, okay, you look euploid, but only with a 95% confidence on one of the chromosomes. If you want a high confidence call, you need to do another blood draw until we are sure that you're getting a result which is a very high confidence result. So here's one of the key advantages of that. The approach works just the same on all chromosomes. The existing techniques, which are quantitative, tend to work well on the chromosomes that amplify reliably 21 and 18 tend to be fairly stable amplifiers. But when you get to small fractions of fetal DNA, the variable levels of amplification in those chromosomes compared to your reference chromosome will tend to cause false positives and false negatives. And if you don't know the confidence in your individual call, you don't know that those are likely to be false positives and false negatives. This is particularly bad when you get to chromosomes like 13 and the sex chromosomes, which have substantially more amplification variability. This doesn't really affect us. The performance across all the chromosomes is the same, and we're increasing clinical coverage by more than a factor of 2. And that's something that's extremely important. We are increasing the number of abnormalities that you can detect non-invasively by looking at the sex chromosomes by roughly a factor of 2. And I've just... Okay, so that's what the data looks like, and I can talk much more about data during question time if you're interested. The green, blue, and red line represent our performance as a function of fetal DNA at 2.5 million, 5 million, and 15 million reads. Now, by comparison, if you look at the hugely parallel shotgun sequencing that's done, at around 8% fetal DNA and less, they tend to have a sensitivity around 75%, and that's with about 20 or 30 million reads. So by using this targeted sequencing and the informatics, at about 5 million reads, we're at an accuracy well above 99%. Now, in yellow are the actual plasma samples that have been run at around 5 million reads per plasma sample, and they are performing just as we expect them to perform. If you then include a confidence threshold, so you only include those samples for which you have a high confidence call, we're on the black line. In other words, touch wood, we haven't had a single sample wrong yet. Now, you never believe anyone who says they have 100% sensitivity and specificity, because biology just doesn't work that way and medicine is too complicated. But because we are able to work out the confidence in the individual calls that we make and request a redraw if we have a low confidence result, we get these very high reliabilities. And that would be only subject to the possibilities of mosaicism, which we should talk about during question time, perhaps. So here's basically talking about the importance of being able to cover all sorts of abnormalities across the 5 chromosomes rather than just trisomy 21, 18, and 13. And if you look at 100,000 patients measured and you're just looking at 21, 18, and 13, albeit with less sensitivity on 13, you're going to see a lot of cases which are not detected by the existing approaches that are out there. And then my last slide is just something which I think everyone should be aware of, which I think the industry is going to be, which is going to be incredibly important to the industry over the next couple of years. The Maternal Fetal Medicine Consortium has just released results on a large study of arrays applied to amnio and CVS samples. And they found that 1.6% of amnio and CVS samples, not in particularly high-risk patients who had an abnormality by ultrasound, but just across the board, 1.6% of samples had a microdeletion or duplication in a clinically significant region. That's a huge number. So we went back to our products of conception samples and looked at over 2,000 POC samples at a granularity of 1 megabase or more. And we found about 1 in 80 samples had a deletion or duplication at a region that was potentially clinically significant. So if you focus on the 10 most common deletion and duplication loci, and you say you can detect at a level of 1 megabase or more, you're capturing about 83% of that load. So that's about 1 in 100 pregnancies. Now, it really depends on what the diseases are. They're not all similarly penetrant, but whatever threshold you use, you are going to be detecting diseases which are way more prevalent than Down syndrome. And this is going to be something that is incredibly important to do non-invasively, because basically everything that you can detect by an amnio and CVS, we would like to be able to detect with the free-floating fetal DNA in the maternal plasma. What does all of this mean? It means that there is a huge need for genetic counseling. We've got 10 genetic counselors in the company right now, and the need for their time with patients grows every day as one starts to get more and more advanced in terms of things that one can measure, especially when you're talking about diseases that are not 100% penetrance, but high enough penetrance that you really would worry. The need for that genetic counseling support becomes paramount.